I'd also like to begin today by acknowledging the Kaurna country that we're on today, and I'm just going to do it in Kaurna language first and then English. So, Nadu Tanpindi, Nadu Kaurna Yantanga, Tekendi. And I'd like to say that I'd like to ask you to join me in acknowledging the first scientists and the people that have walked this unceded land for thousands of years, the Kaurna people. I'd like for us to pay our respects to Kaurna elders, past and present, acknowledging their deep connection to this land and the waters here, and extend this respect to all Aboriginal and Torres Strait Islander people here in the room with us, and all First Nations people that might be here today. Now, it's particularly meaningful that we gather today on World Environment Day, where the official theme is Inspired by Nature for Climate for Our Future, emphasising the urgent need to respond to climate change and ecosystem degradation. Those urgent signals that the Earth is sending us about the fragility of life on our planet. Today's talks will demonstrate that here at the South Australian Museum, we are indeed listening to those signals. You will hear from 9 of our fabulous colleagues here today, spanning research staff and honorary researchers, each sharing insights into their work and how it connects to the environment. All these speakers are connected to the collections and research activities currently undertaken at the museum's world-renowned Science Centre, located just a short distance from here within Adelaide's Cultural Precinct. The museum is currently undertaking an important period of planning for the future, including the development of a new research strategy and collections management plan. These are being shaped in collaboration with an external research collections expert advisory panel, as well as through consultation with key stakeholders such as universities, government agencies, and our honorary researchers. This process ensures that the plans that we make are robust, informed, and aligned with both community needs and state research priorities. I'm very pleased to share that these plans are now nearing completion, with significant contributions from the very people that you'll be hearing from today. Our research strategy will focus on delivering the greatest public value for South Australia, while contributing to national and international knowledge. Over the next 5 years, we aim to sustain and build on our core strengths in biodiversity, earth sciences, taxonomy, environmental change, genomics, and cultural collections. We will also strengthen genuine partnerships with Aboriginal and Torres Strait Islanders and their communities. We will build new capabilities in areas such as climate adaptation, biosecurity, critical minerals, and digital access. And we will ensure that our research is accessible and useful to policymakers, educators, and the broader public. Alongside this, the Collections Management Plan outlines how we will care for, document, and provide access to the museum's collections over the next 4 years. A key part of this work is the state government's new purpose-built cultural institutions storage facility, and that's out at Walkley Heights. At the moment, we're currently moving most of the humanities collections into that place right now. This facility will house collections from the State Library, History Trust, Art Gallery, and of course the museum. Over the past year, our staff have been carefully cataloguing and relocating these collections, and this is a process that will be completed later this year. The facility will provide secure, climate-controlled storage for some of South Australia's most precious items, while also improving access to research and collections. As you can see, the work of our researchers and the collection managers that we have is both extensive and vital. They help us understand our world, guide us through times of environmental change and uncertainty, and ensure that our shared heritage is preserved for future generations. I'm certainly looking forward to hearing what our speakers have to share today, and I'm sure that you all are too. So today's presentation is part of the museum's Sprigg Salon series, which provides audiences with access to the latest thinking around scientific, cultural, historical, and natural world research and discoveries from South Australia and around the world. It is named in honour of Dr. Reg Sprigg, AO, who was a remarkable South Australian geologist who documented the world's oldest fossilised animals in the Flinders Ranges in 1946, now recognised as the Ediacaran fossils. And just as an aside, if anyone has any interest in reading a little bit more about Reg Sprigg, there is a fabulous book called Rockstar that was, I believe, written by his daughter-in-law. And it's a wonderful account of a remarkable life, which also included a little bit of marine biology and the first diving surveys within South Australia, as well as the fossil things. So I would recommend Rockstar. I would also like to thank the company that Dr. Sprigg founded, Beach Energy, for its continued support of this series. I would like to now introduce Associate Professor Diego Garcia Bellido to chair our first session. Thank you.
Thank you, Rachel. Thank you, everybody, for coming today. I hope it's a session that you will remember through the years. It is a brief overview of some of the things we do at the museum. Of course, we do a lot more than this, but we each were invited to choose a few elements of our presentations. To start off our presentations today, we're going to go with who better than the head of sciences at the museum, Dr. Rachel King, who's been a senior researcher since 2009. She works on aquatic invertebrates, as you'll see from her presentation, crustacean in particular. Today, Rachel will present some of her recent work exploring and describing new species of crustaceans from the deep sea and some unique subterranean habitats here in the state. Rachel?
Okay. So as Diogo said, and I repeated earlier, I'm the head of Earth and Biological Sciences here at the museum, which means that I manage the Earth and Biological Science researchers and I represent research at the leadership level within the museum. I'm also a senior research scientist for aquatic invertebrates, mostly marine, but also a little bit of freshwater and a little bit of terrestrial, as we will talk about today. As you may expect, many of us have specialties. And while I am fairly well versed in most marine invertebrate groups, my specialty is crustaceans. You may think of crabs and prawns or shrimp as what you think of when you think of crustaceans. I'm even more specialised and I like the teeny tiny things. Crustaceans are one of the most diverse groups of invertebrates in terms of their body morphology. They can be filter feeders, predators, scavengers, and they live in almost all environments, terrestrial, marine and freshwater habitats globally. The crustaceans that I'm most interested in are the teeny tiny ones that often live in benthic environments, seafloor environments, and they're really important for many environments by turning over organic matter and keeping those environments healthy and clean. For the purposes of this talk, I wanted to highlight some of my work in more extreme systems, and these would be deep sea habitats and groundwater systems. So why is this fascinating to me? Both habitats, they're remote and they're hidden. They're very hard to get to. Deep sea oceanographic work, we're talking anything below 200 metres is considered deep sea, but I'm talking 1,000 metres to 4,000 metres under the ocean is the bit that I'm really interested in looking at. Desert trips to go to look at groundwater through wells and bores, very remote areas, not something that you think about collecting aquatic things so much in deserts, but also can be remote and tough and hard to get to. So these are, so what's happened is we're finding in these areas really hidden communities of invertebrates. And when we acknowledge that almost 90% of our invertebrates are undiscovered and undescribed, these things become really so much more important to us. We're finding these new communities, new habitats with real suites of animals living there, ready to be described, and for us to be understanding what they're doing in the habitats, what their role is within those habitats. And we discover new species and new communities with almost every research expedition that we do. So first, deep sea extremophiles. Life at the bottom of the ocean, it's one of enormous pressure, very limited light, and as an example here, so we're talking lots of things that are happening in the deep sea, gigantism. And though a lot of the animals I look at are less than a centimetre, gigantism for my animals is about that big. That's gigantic. So gigantism, extreme body morphologies, really long legs, long antennae, that sort of thing. That's the kind of thing that is really cool about deep sea systems and the animals that live in them. So as an example, this is an arcturate isopod on the left. It's a crustacean, and it's a very specialised, the back end is on the right, the front end is on the left. You can maybe see a big eye. It's pink, which means in deep, dim ocean, it is almost invisible. It has a nice big eye that it can see. It has long legs to grasp the corals and things that it's living on, and it has front legs that are very hairy that it filter feeds with. These animals, as I said, they're about this big. They're gigantic in my world. And they were first found in the Antarctic deep sea. So their family is called Antarcturidae. But what we're finding is they're popping up in the northern hemisphere, in the canyons and the marine canyons of the northern hemisphere. So we have to really rethink what our origin of these groups, what the evolutionary origin of these groups is. That we're finding that more and more they're globally connected. And that's just bringing up some really interesting stories. We don't often get to go out there, so it's really rare and expensive that we get to go out there and collect. So when we do, it's like Christmas. It's like just people are very excited, and it's really just an amazing opportunity to get samples from some of these places. The other animal here that looks a bit like a trilobite is a saurolid amphipod, flat and built for skittering around and scavenging. Very little is known about their lifestyle. So the other selection, the other habitat that I wanted to talk about was subterranean freshwater systems. And the same animals that I've been finding in marine, their relatives are living in freshwater groundwater-fed aquifers, that's what I'm trying to say. And there are whole new communities that we did not know existed 30 years ago. Australia was not supposed to have the kind of stygofauna that we have. We've just been throwing nets down bores and wells, and there we are. There's whole communities of crustaceans, dominated by crustaceans, living underground in desert systems across South Australia and Western Australia, and also parts of the East Coast. Molecular work tells us really interesting stories of evolution, giving us insights into ancient landscape connections, possible marine inundations spreading species far and wide, climate change sending surface water species underground. We aren't just describing species for the sake of it. Biodiversity work can help underpin connections between regions, determine healthy or degraded habitats, and we can begin to think about how Australian landscapes and their species have changed over time and various climatic conditions. Groundwater in many places like the desert is precious and under threat, and biodiversity data can be a powerful tool for understanding these habitats. So how does this apply to museum work? While I've only talked about 2 habitats, I work across many. I've done terrestrial bushfire recovery projects, and I've also just dipped nets in people's backyard wells at Mount Lofty and found a whole bunch of new species. But we use modern tools, identification tools in our work, leveraging industry and governmental partners for top-tier funding. Species work, or taxonomy, is primarily done out of herbariums and museums. Okay. Our research expertise and biodiversity data makes partnerships across government a no-brainer. I've recently been involved in harmful algal bloom response work. And in terms of legislative frameworks, our data feeds into the Federal EPBC Act and the Biodiversity Act. Some of our groundwater habitats are also threatened ecological communities. Lastly, to finish, we are museum scientists, bringing research information to all our audiences and communities through public programs and exhibitions, STEM education, collection work, and scientific discovery. We bridge the gap between complex research and everyday life to inspire curiosity in learners of all ages.
Okay, thank you so much, Rachel. Let's move on. We have a researcher that has been at the South Australian Museum with us for over 30 years. That's Professor Steve Cooper, principal researcher at the museum and also professor at the Adelaide University, where he uses DNA sequence analysis to explore the diversity, evolution and conservation of Australian animals in general. But today he's going to be talking about the significance of genetics and taxonomy with a research study on South Australian bandicoots. Please welcome Professor Steve Cooper.
Thanks very much, Diego. Can everyone hear me okay? No? I better speak a bit closer. Okay. I've been lucky enough for the last 28 years to be studying this remarkable group of animals that we've found, and Rachel's already mentioned, in groundwater systems across Australia, but particularly in arid regions of Australia. And these animals include arachnids, and you'll hear a little bit more about some of the arachnids that are living in cave systems in Australia, but also a very wide range of different aquatic crustaceans, and even we find diving beetles in the middle of arid regions of Australia. Quite remarkable fauna, and my specialism is to use DNA sequence analyses to explore the diversity and evolution of these animals. And what we've found is remarkable adaptations to living in dark. Many of these animals in very isolated groundwater bodies. And you can see some of these characteristics are they lose their body pigment, they lose their eyes. And how do we do it? The approach that we use is called molecular taxonomy, and we take a specimen, this is a cave cricket, remove, say, a chunk of leg, grind it up and extract DNA, and then we use a technique called PCR, and you've probably heard about it with the COVID method for picking up the virus, to amplify DNA sequences from these different specimens. And then we analyse these sequences, align them, and build trees showing how these different species are related. And from that tree, we can determine whether we're dealing with a different species in that system. And using this approach, we've managed to discover hundreds of new species in these arid aquifer systems in Australia. The taxonomy work, which is what Rachel does, is ongoing, and that takes some time, where we're describing the morphology and putting a name on these different species. So a lot of my work has been on invertebrates, but there's actually lots of taxonomic problems when it comes to mammals as well. And so I've also been doing some work on mammals. I'd like to present the rest of the talk on bandicoots, which is a group I've been studying for quite a number of years, where there's been essentially some major taxonomic issues, and it's been in a state of flux for many years. Currently, according to the Strawn's Mammals of Australia, published in 2023, there's 5 species of bandicoot in Australia, and you can see the distributions of these different species. We have the past distributions in light blue, current distributions in dark blue. So you can see there's been a drastic reduction in the distribution of all these species, and that's happened since Europeans colonised Australia. Just homing in on what's in South Australia. In the current taxonomy, we are considered to have the subspecies Isodon abesilis found into the lofty ranges, and then a different subspecies in some islands off the Eyre Peninsula. Again, they were much more widespread in South Australia in the past, and so this particular subspecies is considered endangered now under the EPBC. So, sorry, I'm getting technical now, because I wanted to show how we use DNA sequences to explore the diversity of species. And we obtained sequence data, and this is a very large study involving a big group of collaborators. And we obtained about 519,000 of these little nucleotides that we could then explore the evolutionary history, the relationships of these different animals, different bandicoots. And generally what we've found is good support for the different species that have been described, except when it comes to South Australia. If we take this portion of the tree, of the relation, and these are individual sequence, individuals that have been sequenced for all these nucleotides. Thanks. What you can see is that the lofty ranges, Kangaroo Island and islands of the Eyre Peninsula, have a species, well, that's very, very closely related to the species that's found in Western Australia, Isoodon fuscoventa. And as of yesterday, this is a very recent result, we have a new species for South Australia, which is the same species that we have in Western Australia, Isoodon fuscoventa, but it'll be classified as a different subspecies in the Mount Lofty Ranges and also on Kangaroo Island and the different islands of the Eyre Peninsula. It's likely to maintain its endangered status at the subspecies level, but I think this just highlights an example of the importance of museum collections in giving us an idea about the historical distributions of species, how they change over time, and also they're a wonderful source of DNA that we can use to develop these complex trees of relationships between individuals and finally resolve the taxonomy. And it really is critical for how we manage these species in the future, their conservation management. So I'll finish on that note, but also I want to just say that this is not just me doing this research. We have an incredible team of collaborators, including my good colleague Terry Bertozzi, who's going to talk a bit later. Both of us have lost a lot more hair and gone grey in the last few years when we first started studying bandicoots about 8 years ago. And thanks also to some critical funding bodies as well. Thank you. I've said enough.
Spot on. Well done, Steve. Okay. Now on to the younger generations. As you can see, not all of us are this old. We're getting new people engaged in what the museum does and bringing a lot of energy with them, as you'll see from Erin. So our final presenter for this first session is Dr. Erin Fagan-Jeffries. Thank you for coming. Erin is a researcher at Adelaide University, where she leads the WASP biodiversity group, and with a lot of effort and a lot of contributions from citizen science. Since 2020, Erin has been an honorary researcher with us, where she works closely with the collection managers from the terrestrial invertebrate groups. Welcome, Erin.
Thank you so much. So yes, as Diego said, I'm an honorary researcher, and that is something I take great pride in. My connection with the museum started in high school. I volunteered in the entomology collection. In undergrad, I volunteered with the education team. In honours and my PhD, I was supervised by the two wonderful researchers you've just heard from. And now as a researcher, I get to still call this place home a little bit, which is very exciting. And I work on wasps. So I want you to just picture a wasp for me. Just take a minute, picture a wasp. Hands up if you are thinking of one of these two things. Okay, quite a few, not everybody, so that's exciting. But this is what most people think of when I say I work on wasps. So on the left, we have the invasive European wasp. It causes lots of damage, it stings, it steals your barbecue meat. On the right, we do have a native species, a paper wasp. This is an Adelaide species. You've probably seen it in your garden. They're not aggressive, but they can sting. So people do have a negative association normally with wasps. But some wasps look like this. This is a beautiful cuckoo wasp, photo taken by my colleague James Dorey. And these cuckoo wasps, I think, are the poster children for wasps. They're beautiful and they're very clever. They actually sneak into the nests of other bees and wasps and lay their eggs inside. So instead of having to find food for her baby, the mum just drops off her eggs and leaves, and her baby will eat the food provided by the other bee or wasp mum. And some wasps look like this. Many, many wasps are tiny, less than a couple of millimetres long. So this is a wasp that's only 2 millimetres long, and it's the kind of wasp that I've worked on for about a decade now. This is a subfamily called the Microgastrini, and they're parasitoids. So most wasps are not social like the European wasp or the paper wasp. They actually lay their eggs inside other insects or spiders. So she will sneak up on a caterpillar, and she will use her ovipositor, which her stinger has evolved into, an egg-laying tube, and inject her eggs inside the live caterpillar while it's wandering around your garden. Her eggs will of course then hatch inside the caterpillar while it's still alive, and her beautiful babies will then eat the caterpillar while it's still alive. Once they're big enough, they will burst out of the caterpillar like a scene from the movie Alien and go through metamorphosis. So just like a caterpillar turns into a butterfly, our lovely baby wasps will make a little cocoon and turn into an adult wasp. Now this might sound a bit gruesome, but they're very, very important for the environment. They control native caterpillar populations, and they help to control pests. So if you've had the white cabbage, butterfly or moth around your veggie patch, eating your brassicas, this little Catessia glomerata is probably also there helping to control those populations. So if you ever see these bright yellow cocoons in your veggie garden, it's made by this little wasp. If you like potato chips, there is a wasp that helps to control the potato moth caterpillar. And if you've had a problem with clothes moths in your house, there's even a wasp for that. So if you've ever seen one of these tiny little black wasps inside your house, it's probably the wasp that's helping to control these little clothes moths. Now we think that most insects in Australia are actually undescribed. They don't yet have a name. We think only 30% of the estimated 220,000 insects in Australia have a name and are identifiable. How do we conserve our local insects? How do we use them if we don't even know what they are? So that's our job as taxonomists, is to find a species and make it identifiable and give it a name. So to find these insects, sometimes we head out in the field to find new species, but a lot of the time we head to museum collections. So these are my students in the museum in Hobart, where we visited to find the local species there, and we have scientists visiting our collections here in South Australia from all over the country and all over the world. There are undescribed species sitting in those collections waiting to be named. But sometimes we get the help from the community all around Australia. So we ran a project working with 50 schools around Australia where we sent the kids an insect trap, they set it up at their school, and then they sent us all the bugs they found, and we helped them name some of their new species. So to finish off, I thought I would introduce you to a few of the species that these students have helped us name. Because naming the species is a lot of fun, but eventually you start to run out of ideas once you've named a few hundred wasps. And it's a lot of fun to work with kids and see what they come up with. So this is Pantelis Darth Vaderi. It was named by Back Plains State School in Queensland, and you can maybe get where they're coming from. They thought the wasp had gone to the dark side, like Darth Vader, because it attacked caterpillars and burst out. Perhaps I got a bit graphic in the presentation to those students, but we thought this was a really creative name for this lovely little parasitoid wasp. This is a Pantelis sinuculus named by Streaky Bay Area School here in South Australia, and often these species names use Latin. So this sinuculus means little bay, and they named it after the local area of Streaky Bay. This is Coccididium melosi heroin. Melosi heroin means honey-coloured hero. And the students thought this wasp was a hero because we know this wasp actually attacks a new pest that's been introduced to in Australia, fall armyworm. So they thought the wasp was a hero for helping the local farmers, which we thought was a really lovely idea for a name. And this is Paraphylax chargrillis, named by Kangaroo Island Community Education because the students thought the wasp looked like had been a bit char-grilled on the barbecue. See the beautiful red and black coloration. So yes, being a taxonomist, we get to come up with names, but my favourite part is actually working with students to help them name their local insects. And we have quite a few left to do, so yes, still lots of work to get all names on all these beautiful insects. That's it from me.
Thank you so much, Erin. Well, thank you, our three speakers to begin with. And I would like to invite the audience to pose a few questions to any of the three, please come up, any of the three speakers. Yes, wait until we actually have the microphone, handle you the microphone so that you can actually be recorded. Any questions from the room? Come on, don't be shy. Thank you. Here, second row. Thank you.
Thank you. This one is for Rachel. Rachel, I'm interested in the limestone coast and groundwater there and the crustaceans that might be there and what might the impact of fracking be on them. Are you able to have you got information about the ecosystem down there and the potential impact of fracking, and how do you convey any information to the government?
So I think our role is in terms of biological diversity. So limestone coasts, we've been looking at some of the springs and looking at some of the groundwater that lives in some of those springs. There isn't that many springs around that also have big communities of crustacean species that I'm looking at. But I think our role is, and it has been across other parts of Australia as well, in identifying that biological diversity and thinking about the species and the way they're related to each other, where they're distributed over the landscape. So that gives information sometimes about connectivity of land, of what is under the water that is underneath the land. There are complex habitats underneath. We don't always understand how connected this spring is to that spring, perhaps. And so by looking at the biological, the species that are living there, we can get more of an understanding of that. I don't have anything to say in particular about fracking, but the work that we do, I guess if there's presence of animals and we know that they're there and they're healthy, that we have more of an idea that that is a healthy environment. It's something that we've had lots of discussions with EPA and DW about maybe them thinking about bringing biological things into their assessments rather than just potentially contaminations, things like that they're looking at right now. So these are ongoing discussions about introducing these other elements, biological diversity, and thinking about that and those connections.
Thank you, Rachel. Another one here in the front. Another one at the front here. Thank you.
Thanks very much to the speakers. My question's also for Rachel.
She does get paid the big bucks.
Yeah, so I've been working as a citizen scientist at Glenelg Beach in relation to documenting the HAB, and I'm aware that there's been an enormous number of aquatic invertebrate mortalities. If you look at iNaturalist, these actually go into the millions, and they're only the observed ones. So there's probably a lot more on the benthic layer that have been not observed or accounted for. So we suspect probably into the billions. So what do you think, Rachel, the long-term impact of that might be, and how long we might take to recover if we don't get another HAB on the back of this one?
Super hard question. It's really hard to say recovery times for these kinds of environments. We don't know a lot about a lot of the life histories of things that live in benthic environments. We do know that the smaller things tend to be more range specific. So we may be getting dark extinctions. We may be getting things that are local, that exist locally, that have died out. But there might be populations elsewhere that might be coming in. It's a really big question, and it's part of the work that we're doing at the moment with DW, thinking about how we can better pull together this data and start thinking about funding some of these taxonomy and discovery programs that think about the cryptic animals, so that we can have these these answers.
Thank you, Rachel. We might have time for one or two quick questions.
Thank you. I'm sorry, I didn't catch your name, but I referred to you as the wasp woman. I'm just curious about the wasp that lays its eggs into a live caterpillar. Just curious to know what the gestation period, how long before those grubs emerge?
It varies for different species. So wasps that lay their eggs in hosts go across the whole tree of Hymenoptera of wasps. But normally it can be a few days to maybe up to a few weeks. I don't think there's many species that stay inside the host for much longer than that, but I'm looking at Ben, he's nodding, yeah. Yes, so normally a shortish time period while they're eating till they're big enough to be able to metamorphise. Yeah.
We got one last question. Really quickly. Thank you.
Can I do two sub-questions?
Well, we'll try.
The question relates to caves and what's in the cave. Do they have mygalomorphs down in the caves as well as the arenaomorphs, the type of spiders? I'm quite curious because you were talking about the huntsman sort of ones.
Yes, stay tuned on that question. You're absolutely right, they do have megalomorphs, and you'll hear more about these cave spiders from Jess very soon.
And one other second part of the question to Rachel. Rachel, with the rate of decline of the depth, as the depth gets depth increases, if there's a very sharp drop-off, how are the populations likely to be affected compared where there's a gentle drop-off to the deeper waters in terms of populations and their risk? If you have a very deep drop-off, like 0 to 2,000 or 3,000 metres versus 0 to 200 metres over a longer distance.
I think that would be very hard to say across a broad group of things. You're going to have specialists that will be at certain depths, certain pressures. So I don't know that's. I think more the animals will have a niche that will be their habitat, and I'm not sure that a depth gradient would. I don't know. I have to say I'm not sure.
Thank you very much for those questions. And just so that we can keep it on track, I'm going to invite everybody to thank our three speakers and move on to the second session.
To start our second session is Dr. Mark Stevens. Mark is a senior research scientist in terrestrial invertebrates at the museum and has been around at the South Australian Museum since 2008. His particular focus looks at changes in biodiversity over time with changing climates. Mark will bring the fantastic world of springtails to life by taking you into his research from Antarctica to the Gondwanan forests of Australia.
Yeah, thanks, Rachel. Welcome, everybody. So I work on springtails, and I guess you're asking what the bloody hell are they? And it's times like this, especially given his very significant birthday, that
There is a huge and varied population of animals living on the land with bodies that are a little different from those of their ancestors who lived in the sea so long ago. And they are extraordinarily successful. Some are the most numerous of all land-living species, but we seldom see them. This pin will give you an idea of why. They're tiny. This minute little creature is a Sprigg animal. It's less than 0.5 millimetre long, the size of a full stop.
So, David, so although most are less than 2 millimetres long, they are also, the ones that I study, are actually the giants. They are the largest in the world, and get ready for this, they are 17 millimetres long. You got to be impressed. And so the group of these giants, the superfamily that I've just recently published on, it's taken 25 years to collect the animals necessary for that globally, so that we could tackle a group which is shown in the orange bars on that genetic tree. So we've used DNA, and you've heard some of that from Steve about how we utilise that to understand the relationships of species. And what we were able to do and I thank the Waterhouse Club in Adelaide for this, because there was a critical species of the family that we needed to be able to solve this mystery of these groups which don't form a natural group. And what we did, we were able to dissolve that subfamily, Euchidnurinae, and erect 2 new subfamilies. That doesn't happen very often, but what we found that was really surprising in this group was that both of these were southern hemisphere, dominated by southern hemisphere species. Again, not very often, and this is the first time it's been recorded for springtails. And interestingly, the Acanthonuinae, equally hard to say, so is dominated in forests. It's just found in New Zealand and Australia. Forests like this, Nothofagus, these are the ancient forests in Australia that mimic what the forests were like during Gondwana, when Australia, Antarctica, South America were all joined as a supercontinent. And in South America and Tasmania, these forests still are remnants of that time. I draw your attention to one at the bottom, which is the deciduous Nothofagus. It's adapted to cold climates, very cold in alpine regions, but you don't get as cold as Antarctica. And how does that relate? Well, there are springtails there as well. But what I'm relating this to are the fossilised tree stumps in Antarctica, just south of this region in Antarctica where I've spent quite a bit of time. These represent new species, but also species that are still found in Tasmania, in Queensland, and in South America. And what does this do? It allows me to have a drink. What this tells us is the climate in Antarctica, what that was like 10 million years ago. These fossils give a picture of the ecosystem, the animals, the plants that are found there, that still exist in South America and Australia when they were joined as Gondwana. So it's recognising the connections that Antarctica had to these land masses. And the work that I've done in and around Antarctica is use DNA to understand the connections, so the connectivity of the islands around Antarctica, their connectivity to the continents, Australia and South America. And in some cases, they're very restricted, only found on one island, but in other cases, because we're using DNA, we're able to map the connections that are occurring between islands. But when we look at Antarctica without ice, and the browns and the greens are all above sea level, and each of these bubbles in Antarctica represent distinct species, not found, but only found in each of those bubbles. They're not found, no sharing between the bubbles, and these species are not found anywhere outside of Antarctica. So they've always been there. Now, how is that possible? So what we had to do was come up with a theory. How do things survive in Antarctica during glacial periods when there's a lot more ice and snow around? And what we do in glacial periods, we were able to show from studies that have been done in alpine regions, South America and New Zealand, showing how animals track advancing ice and also recede with that advancing ice. So that advancing ice, so that's an environment that is favourable for these animals. It's the interface between the ice and the land, where there's meltwater, which is critical for the survival of these animals. And although most of you are probably pretty fond of vertebrates and penguins and things, some of them are okay, I guess. Some of them are a little bit scary. But I hope that you will be like me and think that these are really the fantastic beasts And I always call on cartoon Diego to help me out in situations like this. You're going to hear later today talking about his Cambrian fossils. But what I work on the evolutionary success stories from the Cambrian period. These are the animals that survived and we have with us today. And I get to privilege of working with colleagues, students, volunteers and research honorees on a much larger suite of animals, invertebrates at the museum. Thank you.
Thanks, Mark. A reminder, we're just going to keep the questions until the end. Continuing the terrestrial invertebrates theme is our next speaker, Dr. Jess Marsh. Jess is a research fellow at Adelaide University and an honorary research associate at the South Australian Museum. Jess will talk about her latest adventures surveying remote caves of the Nullarbor and some of the weird, wonderful, imperilled cave-adapted spiders and species that the team have found.
So, hi everyone, my name's Jess Marsh, and so today I'm going to be talking about our expeditions into the Nullarbor where we're documenting the invertebrate life there. I just wanted to start quickly by acknowledging the traditional owners of the land we meet today, the Kaurna people, but also the traditional owners of the Nullarbor. It's been a real privilege during these surveys to work with cultural monitors and traditional owners, and I pay respect to their elders past and present. So the Nullarbor is a globally significant landscape. At over 200,000 kilometres squared, it's the largest cave region in Australia and the largest arid limestone cave region in the world. There are thousands of caves under the Nullarbor, ranging from giant, big, cavernous chambers that you could park a jumbo jet in, through to giant caves with crystal clear blue lakes, and then you get down to the tiny little squeezy crevices, which are kind of the fun ones. It's also a land of extremes and contrasts, so it is a very dry place. In the summer, it gets very hot. In winter, it gets very, very cold. However, underground in the caves, it's a much more stable climate. So typically, it's around 18 degrees Celsius, and we see about 70 to 80% humidity. It is also a centre of importance for biodiversity, so we know that there are animals there that are found at the Nullarbor and nowhere else in the world. One of them, which is this spider that you can see on the right here, is, as the question was asked earlier, this is a megalomorph spider, cave-adapted megalomorph spider, which is the group that also includes funnel webs, tarantulas, trapdoor spiders, etc. This is the only cave-adapted megalomorph spider in Australia. It's called the Nullarbor giant cave spider. As the name suggests, it's pretty big, so about the size of my hand. It's lost its eyes, so it lives underground in the dark. It's got no eyes, and it's got very elongate limbs, perfect for sensing its way around in the darkness. Now, unfortunately, there are also a number of threatening processes that are impacting the Nullarbor, ranging from feral animals, weeds, mining, development, human impacts such as collection for the pet trade, and of course, climate change. So what we know about the biodiversity of the Nullarbor is really just the tip of the iceberg. Why is this important? Well, it's very difficult to protect species and protect habitats important if you don't know what's there. So in my project, I've been working with a great team of people to go out to these remote caves, to go and try and work out what is living there and to document the species. So on the left, that's me in my happy place, covered in dirt, deep underground, looking at spiders. The top middle image, I don't know if you can see, I'm a tiny speck in the middle. So some of the caves we work in have some pretty exceptional heights. So this was a 35-metre drop that I was abseiling down. On the other end of the spectrum, there are also some incredibly tight squeezes. So my colleague here, Steve Milner, is coming out of one of the squeezes, and we spend a lot of time wriggling through holes. Before I started serious caving, I would have said that I was probably a little bit claustrophobic, probably a little bit scared of heights. I've now come to realise that if there's the promise of a large spider on the other side, I can get through anything. I know that is most people's worst nightmare, but it takes everyone. The team I'm working with has made this all possible. So we work with some exceptionally talented cavers. exceptionally talented biologists and also with traditional owners. So we're working together to try and document this life. So we've done one survey a year so far. We've got more to come. And to be honest, the findings we've made so far are exceptional. This is just a small snapshot of the species we've found. Most of these species here are only known from single caves. All of these species here have lost their eyes. Not a pair of eyes, not a set of eyes in this whole thing. These cave-adapted species are typically incredibly rare, so I feel really privileged to be able to find this. I thought I'd just talk very quickly about two of my favourite species. So, the first one is this wasp. Troglopampilus muricicatus. The paper in which we described the species and the genus was out a couple of months ago. It is the only cave-adapted wasp known in the world. It's lost its eyes, and as you can see from the image, it's got very long antennae and very long limbs. I will quite literally never forget the moment that I was crawling down a very rocky, uncomfortable passage and saw this animal. As soon as I saw it, I knew I was looking at something special. It's only known from dead individuals, so we haven't been able to find a living one, and this species could well be extinct. It's only known from a single cave. We've got a lot more research to do, though, to try and work that out. The second species is probably one of my favourite. This was the first species of the Nullarbor fragile cave spider that is known from South Australia. This little juvenile here is quite small. The adults are big and leggy, very pale. This species is only known from a single cave, and unfortunately we've documented a lot of threats to that cave. The biggest threats, foxes, and there was a lot of evidence of decline of populations, so a big concern. This little juvenile has actually made its web inside a fox den. So my passion is taking what we've learned on these surveys, bringing it all together and turning that into action to try and protect these places. For me, I think one of the key ways of doing that is collaboration and knowledge sharing. So what we're doing first, I'm assessing all of these species under the EPBC Act or the IUCN Red List for extinction risk. By listing species, we can protect them. We're identifying biodiverse cave where we've got multiple cave-adapted species in a single cave, because that way you get your best bang for your buck for conservation spending. You can protect whole suites of species if you protect that cave. Thirdly, I'm working with traditional owners and ranger teams to co-design and implement conservation management actions to protect these caves. Finally, we're sharing findings. The Nullarbor is not just a boring place that you drive through to get to WA. It's an incredibly remarkable landscape. The Nullarbor is a massive area. There are thousands of caves, many, many species that we need to protect. I feel like bringing together different people from different cultures, different backgrounds, different expertise, bringing together people who care for this place, it's a really powerful way that we can try and protect these species and this remarkable landscape into the future. Okay, thank you.
I'd like to invite all the session 2 speakers to come up to the front to take questions from the audience.
I'd like to ask, which applies to at least 2 of you, in the case where you've got invertebrates without eyes in caves and groundwater, etc., how do they feed if they can't see what they're chasing?
Yeah, that's a really good question, and something I've got some living individuals of eyeless spiders that I've been keeping as scientific research specimens to observe, and a lot of these cave-adapted spiders have really long sensory hairs. So we think they're detecting prey by vibration mainly. There might also be some electroreception, so they have hairs that detect electric currents. I know that when I put prey into a spider's tank, the spider will notice that prey from about 10 centimetres away, and it will go to it if it's hungry. So yeah, it's a really good question, and lots to find out about these animals.
Thank you.
Hello. I sense from your interest in tracking down all these variations of species, getting into subspecies and discovering so many, that there's an underlying sort of principle there that you want to preserve them all. Would I be right? But that's not the question. The question is, does there come a stage in preservation of species where mankind could say, some species we should wipe them out and make them extinct deliberately? I'm thinking of the Anopheles mosquito, which is responsible for so much human misery. Now, if any of the four of you had the ability, the power to do it, would you agree to the deliberate making of extinction of the malarial mosquito? It's a general principle, of course, but that's not a bad one to start with.
I'll have a little go at that. Firstly, we know that a lot of invertebrates, and other species, the network of connections into this food web are very, very complicated. So I think of it a bit like a net. You've got a net of biodiversity. You lose individual species, you get holes in that net, but it still captures things, still holding strong. If you lose more and more, you get bigger holes and it frays and things start to collapse. And so basically, we do not know the role of most species in that net, and it's very difficult as humans to pick and choose and then to be able to predict what the ongoing outcomes of that are. I think, it's a very, very complex world. We're losing biodiversity at a rapid rate, especially with Australia's invertebrates. So it's estimated that over 9,000 invertebrates species have been lost since colonisation.
I think that you're not answering the question. I'm asking you a specific case. I'm giving you a specific case. You're worrying that the Anopheles mosquito removed it might leave some embarrassing or unwanted holes in the web. But would you, if you had the power to do it, could you save mankind from the enormous amount of misery that creature causes?
Well, that's a very difficult question to answer, because if you could say point blank this would save mankind by removing the species, and we could tell that it wasn't going to cause other implications on track, then who wouldn't choose to save mankind? I think the difficulty is we can't say that we can save mankind by removing this thing. We also don't know the implications. So it's a good philosophical question to think about, I think, but a very complex one, highly complex.
We've got three more presenters in this next session, and leading it off is Jacinta Kulmatri, and Jacinta is an Adnya Marana and Ngarrindjeri woman and the Research Fellow for Aboriginal and Torres Strait Islander Culture and Art at the South Australian Museum. And Jacinta will talk about the depth of Aboriginal history and how Aboriginal knowledges are far deeper reaching than most people think. Please welcome Jacinta.
Hello, I'm Jacinta Kulmatri and I'm Adimatna Ngarrindjeri woman. So this is my country. The Adimatna country is the Northern Flinders Ranges. Northern Flinders Ranges, and Ngarrindjeri country is in the Coorong. As a visitor to Ghana country, I also want to recognise Ghana people's continued connection to country and environmental advocacy. Being World Environmental Day, it's important we recognise the deep and continuous connections that Aboriginal people have with the world around us. For Aboriginal people, every day is World Environment Day. My research background is a little different than everyone else's. I haven't worked at the museum in a research role for as long as everyone else. I am an archaeologist and I have worked with my own Aboriginal community and other Aboriginal communities to highlight the depth of our knowledge. I've also been incredibly passionate about helping young people feel proud about their culture and for them to recognise that we are the first scientists of this country. Because as part of Australia's colonial history, being proud about our history and connection to country is something that we have been told we should be ashamed of, and we were forcefully restricted from acknowledging and even continuing this connection. As the first scientists of this country, we've had a long time to understand how the world and our place in it worked. We've had at least 60,000 years worth of time. 60,000 years is a very long time when you consider the history of the world, especially human history. As each square appears, I want you to think about 1,000 years passing. I want you to think about the fact that the first fleet, those British ships that were carrying convicts, with the aim of colonising Aboriginal land did not even arrive more than a quarter of one of those squares ago. I want you to think about Vikings not even making up 2 of those boxes. Think about ancient Egypt barely covering the top line of those boxes. 60,000 years is a very long time, and a lot happened here. We witnessed the climate changing dramatically, sea levels rising and animals becoming extinct. So what were we doing all of those years? Were we wondering about the land? Were we collecting food and hunting animals opportunistically? We weren't. Aboriginal people survived but also lived and loved life during this time. Over those 60,000 years, we were using scientific practices to better understand how to live with the country, animals, and with each other. And I understand that I say that we were the first scientists and used scientific practices to a room full of people who are most likely scientists who gained their qualifications in a lab by writing papers. But the way we actually lived in this place required the use of scientific practices. They may not have had peer reviewers, but they did recognise the impact of their actions. Aboriginal people lived collectively, creating laws and structures that function as our own peer review. We formed our own questions, which are all tied to ensuring that country and people live sustainably. We used observation as a key practice, and through observation, we came to understand how influential our behaviour was, and that in order to care for country, we needed to recognise our role in country. We are a variable we can control. We do not own country, we are part of it. We see this through the use of totems, and our totems are animals, plants, and other beings in the world around us. Totems serve a very important purpose, and that purpose is to ensure that these beings remain protected. Many people probably understand the very basic principle, and that is that we don't eat our totems. Totems are sacred, and by tying ourselves to one, we know there's always someone who is thinking about their safety. We also form complex kinship structures that are still in use today. These structures were vital to our survival and ensured we weren't marrying and having children with people who we were too genetically close to. This is a deep understanding of genetics that cannot be understated, particularly when you consider the level of inbreeding that occurs within royal families across the world. Being able to understand that we were stronger and healthier by forming these structures is a scientific breakthrough ahead of so many cultures across the world. We value community strength as opposed to familial power. And while we may believe that these understandings about the world are new or are overstated, we have a lot of evidence showing the depth of our cultural continuation. Across the continent, we have stories that go back as far as 30,000 years minimum. Some communities have records of volcanoes erupting in the past, animals that became extinct, and just about every coastal nation have stories recalling the sea levels rising after the last glacial maximum, the time when glaciers were at their peak, around 20,000 years ago. The ability to remember these moments in history means that our knowledge is much deeper than most people imagine, and there is so much more knowledge we developed that I couldn't possibly capture it in 5 minutes. What I hope you can take away is that just because science didn't occur within a lab doesn't make it any less scientific. Aboriginal people lived here for millennia, and it wasn't by accident. It was through a deep understanding of country, something we were told we weren't allowed to be proud of, something most of us were forced to forget. So I think it's important that we make sure we respect Aboriginal history, honour country, and we can do that by recognising we were the first scientists of this country. Thank you.
Thanks so much, Jacinta. We'll be having question time at the end of the 3 talks as before. Our next presenter is Associate Professor Diego, I'm not going to say it in Spanish, Garcia Bellido, but it should be Bellido, I believe. Diego has held a joint appointment at the South Australian Museum and also the University of Adelaide, now Adelaide University, since 2018. And where his research is focused on the early evolution of animals from around only 500 million years ago. Today he will present on the latest discoveries and significance of the Ediacara biota of the Flinders Ranges and the Cambrian faunas of Kangaroo Island. So please welcome Diego.
Thank you, Steve. It's a pleasure to be talking to you today. Thank you for inviting me. And I want to start by acknowledging that we're meeting in Ghana land and that the work that I do is in Adnyamatna country. So I just wanted to time travel back about 500 million years to try and give you a brief overview of what this continent, in specifically this state, looked like 500 million years ago. So when is that? Because 500 million years is hard to grasp, $500 million or $1 billion. So the planet is about 4.5 billion years old, and we've got most of the record is just basically stromatolites, those organisms in the middle. These are cyanobacterial fossils. And then we've got a snowball Earth, a moment when the planet froze over at about 700 million years. And soon after that, we've got a fantastic record here in South Australia. In particular, the two fossil groups or fossil times windows that I'm going to be talking about are the Ediacaran and the Cambrian. So that is when. Where do we work? So first, we work up in the Flinders Ranges, Adnyamatna country, in the Ediacaran fossils of Nilpena Ediacaran National Park in the western side, which was just established about 3 years ago. And then we also work in Kangaroo Island. But those rocks are slightly younger, and the place where we can actually study them that have fossils is in the north end, just north of Kingscote, northwest of Kingscote in Emu Bay Shale, which is a type of rock which preserves the fossils. So what do we look at and why do we look at it? Well, this is a reconstruction of the Ediacara biota, as I said, 555 million years ago, so that's about the age of these organisms. Where this is a shallow marine waters that were covering the northern, what is now the northern Flinders Ranges, and the Flinders Ranges in general, and actually even the Adelaide Hills. They're cryogenian, they're about 700 million years, so slightly older than this. But in the Flinders, we've got some fossils in them, and these are things like Dickinsonia up in the top, which is flat, probably about 2 millimeters thick, but reaching 1 meter in length. We also have clear evidence of some organisms, probably among them the most complex of the time, and they're scratching the seafloor, feeding from that microbial mat that was covering the seafloor, where Australia at the time was connected to Antarctica, but we were on equatorial waters, so as you can see, the continents keep moving up and down. We also have Spriggina, which is this one here with the, let me use the pointer, with those units in the body, whether they are segments or not, still being debated. And then we've got things that have no similarity to any living organism today. They have this triradial symmetry like Tribrachidium, this one here, those 3 branches, okay? So one of the things we did about 9 years ago is we invited South Australians to choose a state fossil emblem. And how many people know, raise your hand if you know, which one is the state fossil emblem for South Australia? Good, a fair few of you. Well, it's indeed Spriggina, named after Red Sprigg, who discovered the Ediacara biota this year, 80 years ago. So we're celebrating the 80th anniversary of the discovery of the Ediacara biota. It's an important year, and hopefully the museum gets to organize another Sprigg event celebrating this anniversary. So what we're doing is not only describing the new species, but we're trying to go the next level and applying the latest technologies to our understanding of how these organisms grew, what they might be related to, etc., etc. This is work from one of my PhD students, Tori Botha, using laser scanning, micro laser scanning, then utilizing geometric morphometrics to try to understand how these different organisms, and Eondromeda, but also Tribrachidium, to try to place them into the tree of animal life. The next one that I wanted to talk about, Emu Bay Shale, about 512 million years ago. This is a mural that was, or a painting produced by Peter Tressler, which has just been turned into stamps earlier this year. You can still buy these stamps in Australia Post. And it represents a number of the components of those first fossils of animals that we've got in Kangaroo Island. It's the only place in the southern hemisphere that preserves soft parts. Normally, as you know, the fossils are bones and shells and exoskeletons. So what do we have here? We've got Anomalocaris, the giant predator of the Cambrians. It would have been the equivalent to modern-day great white sharks in terms of its apex predator position in the trophic pyramid. Then we've got things like Redlichia. Redlichia is fully mineralized, as you can see here, very hard exoskeleton made of calcium carbonate, and he was also a predator, and we have evidence of that because we've got other Redlichias that have been attacked. You can see the bite mark that's been chomped off this particular specimen, and it survived and actually healed that injury. We also look at the life cycles of some of the other organisms, like Stangia bilobata here. We look at how they grow. We try to study, are there any things that we see in this growth pattern that is different from the arthropods we see today? We also look at their molting patterns, again, behavior. So we're looking not only at the shape of organisms, but how they live their lives. In this case, organisms, arthropods in particular, need to go through molting in order to grow. And then we also look at their guts. So the gut is normally not preserved. This is not the case in Emu Bay. We do have internal organs preserved, and in this case we look at the trilobite guts. But we also look at some of our family members. In this case, another chordate, Nesanectris. It's our closest representative in the Emu Bay Shale. And of course, this is not done in a vacuum. We are part of teams. This is my Ediacaran research team here at the museum with colleagues from North America, Canada in particular. But this is done through the help of many, many volunteers. We don't do this by ourselves. We have volunteers, especially from groups like the Waterhouse Club and with support from the Flinders Ranges Ediacara Foundation that are helping us with excavating those fossils, but also the Australian Research Council and NASA. And the Cambrian research team, a similar thing happens. We're still researching the Ediacaran fossils. We're no longer collecting down there, but this is a multi-institutional effort, like most of science is done nowadays, including national ones like University of South Australia, UNE, and Macquarie, but also the Natural History Museum in London. And again, we use the financial support from a number of sources, not least, obviously, our universities present, and also international, like National Geographic, the Spanish Research Council, and Australian Research Council. And with that, I wanted to thank you for listening.
Thanks so much, Diego. And spot on time as well. Our final presentation continues the paleontology theme, but this time a little closer to the present. Professor Mike Lee is a senior research scientist at the South Australian Museum and an honorary professor at Flinders University, studying evolutionary biology and paleontology. His research looks at how organisms are related to each other using both anatomical and genetic information. Today, Mike will talk about crucial fossils, including the giant rainbow serpent. And sorry, I should have moved on. Including the giant rainbow serpent Wanambi from South Australia, and how they've shed light into the origin of snakes. So please welcome Mike Lee.
Thanks, Steve. So we're going to time travel forwards now 550 million years to the last 50,000, because nothing interesting happened in those 550 million years. Don't mention dinosaurs. So I'm going to be talking about a snake that lived about 50,000 years ago in South Australia called Wanambi, and that's on display on the top floor of the museum. Who's seen it, by the way? Great, wonderful. So it would have been about 5 meters long, and it lived in the late Pleistocene. As I said, it lived to within 50,000 years ago, so it would have overlapped with the first Australians. And fittingly, its scientific name, Wanambi, derives from a word found in many of the Aboriginal languages in Central Australia, which have a word for fossil snakes which lurk around waterholes, and the word is something like Wanambi or Wanampi. And this fossil was discovered in the 1980s by John Barry. Now, John and Julie are unsung heroes of South Australian science. They have been excavating fossils in the Narracourt southeast region for about 50 years, sorting through them, describing some of them, and many of those fossils are in our museum and have shed really, really important insights into the history of life. And John is also a gun artist, and his reconstruction of Wanambi is in front of you there too. So he not only found it, but he also drew it and reconstructed it and described it. Now at the time, so Wanambi is a bit of a celebrity snake. It's got a building named after it near Narracourt. It's got a street named after it. It's been on the cover of Nature. It's been on the cover of The Australian. So why is it, the Taylor Swift of snakes, if you like? It's a type of snake called a matsoid. So it belongs to an entirely extinct group of snakes. But these matsoids were originally thought to be not that interesting. They were thought to be similar to modern constricting snakes like pythons and boas. So, a little bit different, but not that astonishing. However, our research showed that they're nothing like that. They're actually much more primitive than any snake alive today, totally unlike anything around today. They're incredibly ancient and incredibly primitive. And so, as most of you know, snakes evolved from lizards. They're essentially legless lizards on steroids. And matsoids retain many of the features found in the earlier snakes. Some of the relatives of matsoids, the relatives of Wanambi from South America, still retain little tiny legs, little fully formed limbs. We don't have legs from Wanambi, but we do have a large preserved pelvis, which suggests that it probably too retained legs. And also, if you look at Wanambi's skull, it's also got some very primitive lizard-like features. For instance, its orbit, the eye socket is completely surrounded by bone, which you don't find in living snakes today because their skulls are famously reduced. And their jaws, snakes are famous for having very flexible jaws. Wanambi has some, but not all of that flexibility. So in many ways, they're sort of like missing links, if you like, between lizards and snakes. And so this means that they can shed light on why perhaps snakes lost their legs, right? What ecological factors drove them to becoming long and limbless? Did they lose their legs for worm-like burrowing, or did they lose their legs for eel-like swimming, perhaps? So we took a new angle on that. It turns out the body skeleton of Wanambi isn't that informative. If you think of a water python skeleton or a skeleton from a woma from the arid zone, their body skeletons are really similar. They don't tell us that much about their ecology. So we CT scanned the skulls of Wanambi and its older relative Yalunga from Queensland, and we looked at their inner ear. Now, you might laugh, snakes are famous for not having ears, but that's a bit of a myth. They don't have external ears, but they do have this system of canals and tubes in the inner ear near our brain case that we have and indeed all vertebrate animals have. And those ears, those inner ears, the semicircular canal system, is really tailored for the way the snake holds its head in its environment, which of course then reflects the snake's environment. And it turns out that when we CT scanned Wanambi and looked at its inner ear, it showed that it probably had both burrowing and swimming habits. A bit like this snake up the top there, which burrows and swims and has, it's called a dog-faced water snake, and it has the delightful scientific name of Cerberus. If you want to get into details, the semicircular canals of burrowing snakes tend to be narrower, and so Wanambi and Yalunga have this sort of narrow semicircular canals, but the big blobby bit in the middle, the vestibule, tends to be swollen in a particular way in aquatic snakes, and those matsoids also had that sort of swelling. So it's a bit like, everyone's correct. Now, snakes are also unique amongst reptiles in that the brain case is completely enclosed. The brain case completely encloses the brain. So if you have the brain, you can generally make a pretty good inference about if you have the brain case, you can make a pretty good inference about what the brain looks like. So we're at the moment in an ARC project to map the match between the brain case and the brain, which bits are closely matched and which bits are loose, so we can infer the brain of fossil snakes from the brain case alone and ask the question, was Wanambi, did it have a swimming brain or a burrowing brain? Now, the last thing I'll talk about is a few other fossils which shed light on this burrowing versus aquatic idea. And this is a fossil that my colleague and I described from the West Bank near Jerusalem a few years ago, a thing called Pachyrhacus. So this is pretty much the most primitive snake known. It has legs, it's even more primitive than Wanambi, and it's clearly aquatic. It's got a ribbon-like body like an oarfish, and it's got heavy vertebrae like a dugong. So that, to us, sort of tilted the needle a bit towards an aquatic origin of snakes. Then, of course, we looked at what lizards were most closely related to snakes, because we know snakes evolved from lizards. And it turns out that none of the legless lizards around alive today are actually closely related to snakes. Their DNA is completely different. It turns out the closest lizards to snakes are mosasaurs. So we worked on mosasaurs before they became trendy with Jurassic Park. Mosasaurs have teeth in the roof of the mouth to help ratchet down prey, just like snakes do. No other lizards have that. Mosasaurs also have some unique articulations in the jaw joint, which show hints of evolving towards the snake-like jaw flexibility. So they did have some amazingly snake-like features that were completely unexpected. And our biggest achievement was actually publishing this lunatic fringe idea in Nature. And since then it's moved from the lunatic fringe to, I would say, an important minority view in terms of snake origin. So we're progressing. And of course, all this research would not be possible without the wonderful volunteers, collaborators, and funders who have helped. So thank you for listening.
Thanks very much, Mike. I'd like to invite all the presenters back again and invite you all to ask some questions of the presenters.
If the brain is completely enclosed, does that mean it doesn't have a spinal cord?
Oh, there are exits for nerves and veins, so it does have a spinal cord. But it means that, like our brain, our brain is completely enclosed in bone, whereas if you look at the brain of, say, a lizard, most of it's actually not enclosed in bone, it's open. So if you have the brain case of a lizard, you can't really make strong inferences about what type of brain it had. Whereas if you have the brain of a primate like us or a snake, you actually have a really, really good idea of what its brain looked like. But the point is, though, you need to map where the brain case fits tightly against the brain and where it's loose, because your inferences are strongest where there's a tight fit and they're weakest where there's a loose fit. And spoiler alert, the key kind of parts of the brain which tell us about ecology are unfortunately the bits of the brain case which have the loosest fit.
I'd just like to ask, in your Aboriginal collections and Aboriginal research, do you do any work on Aboriginal astronomy? And if not, why not?
I think there has in the past, but not.
Because there is a new book out recently in which a astrophysicist has got together with some Aboriginal people and looked at Aboriginal astronomy and found some very strong connections with recent research, if you like. So
I think,
it seems to me to be an area that might be worth exploring. Thank you.
One of the things about astronomy is that there's a lot of sacred knowledge, so a lot of secret knowledge. So when we do actually talk about it and if we present it, we have to be very, very careful because we could be sharing information that shouldn't be shared with the broader community. Yeah.
Diego, you've got the Ediacaran, you've got the Cambrian in South Australia. Is there any place, do you think, in South Australia where you could see the end of one and the beginning of the other?
The what, sorry?
The end of the Ediacaran and the beginning of the Cambrian. How much of a gap do you have?
Yeah, we believe, so we've got, thank you, Stephen. We've got a big gap between the top of the Rawnsley quartzite and the overlying Urutana, and then in sometimes the Parachilna formations. We believe there's at least 10, 15 million years missing. And this is not only in Australia, this happens around the planet. It happens in the Grand Canyon, for instance, a very famous hiatus. It's what we call the Great Transgression. It apparently, most of the planet, during the end of the Ediacaran and beginning of the Cambrian, the sea receded, and then in the early Cambrian it went up again and it inundated all of that and began the record in the when there's erosion you cannot have fossilization. So with a few exceptions, Namibia being one of them, you can basically you jump from the top Ediacaran to say 5 million of years into the Cambrian. So it's I would love it, yes, but unfortunately I think it's missing. As far as I know, it's missing, and it is missing in the Northern Territory as well. So it must have been the whole continent, which started in Western Australia, the whole band, the coastal band, the sea receded, and then it only came back up into the Flinders Ranges and its extension into the Northern Territory at about 520. So we've lost the Cayman explosion and the extinction of the Ediacara biota.
Okay, if there's no more questions, please. Oh, we have another one from Steve at the back. Very quickly.
Where did the sea go?
Well, we know from glaciations that they can produce a good 120, 150 meter drop in sea levels, as we had at the last glacial maximum 12,000 years ago, and is recorded in Aboriginal stories. So it is a possibility that there could have been some massive glaciations having to do with the cycle of the sun and not the snowball Earth level, but yet that it would have created this major transgression followed by regression of the oceans across basically the whole planet.
Okay, now if there's no more questions, please join me in thanking Diego, Jacinta and Mike for some fabulous talk. Now, I'll pass over to Rachel, who's going to give some closing remarks.
Thank you, Steve. Just to wrap up, today's Sprigg Salon has taken us on an extraordinary journey across the breadth of scientific discovery, from the hidden biodiversity of deep oceans and subterranean caves to the genetic secrets of Australian fauna, and the ancient stories written in fossils and country. Appropriately, as we mark World Environment Day, today's talks have reinforced the importance of understanding and protecting our planet. Our presenters have highlighted not only the diversity of life, past and present, but also the vital role research plays in conserving ecosystems, informing environmental stewardship, and deepening our connections to the natural world. Each talk reminded us that discovery is ongoing, whether it's identifying new species, uncovering evolutionary relationships, or valuing knowledge systems that span tens of thousands of years. As we close, we are reminded of the spirit of Reg Sprigg: curiosity, persistence, and a commitment to expanding human understanding. The work shared today reflects that same spirit, showcasing South Australia as a hub of world-class research and collaboration. On a day that celebrates our shared responsibility for the environment, it is inspiring to see how science, culture, and community come together to help shape a more sustainable future. Thank you to all our presenters for their engaging insights, and to our audience for your enthusiasm and your thoughtful questions today. We hope that today has sparked your curiosity and inspired you to continue exploring and caring for the remarkable worlds around us. Thank you.